it is very appropriate that Don should be interviewed here because he began life as a mycologist, a grown-up mycologist here in Puerto Rico, but Maya Glass.
Yep.
Okay, so tell me about your family. Did you grow up on a farm?
No, I grew up in a small town in Ohio.
Okay.
In farm country.
Well, that's where the farm came in.
And my mother's parents lived on a farm, though we didn't spend a lot of time there, but they lived on a farm. And my father and mother lived in town. My father was a car mechanic and ultimately owned a business, and I had to work with him in the car business.
So you can fix cars?
I could in the olden days.
I had no idea, Don. Hidden talent.
Hidden talent, yes. Yeah.
And you had 2 brothers and a sister?
I have an older sister and 2 younger brothers. And in our family, where my parents were high school graduates, but my father was very insistent that we go to college and that we do the best we could with our lives. And so my sister went to a local college, became a biology teacher, and later on a librarian. One of my younger brothers is a designer, or was a designer. He's retired now, designer of industrial products. And my youngest brother is a physician.
Wow. So kind of biological, several of you.
Yeah.
Yeah. So did your father live to see you at Harvard?
No,
what a shame.
Yeah, he died in 1971, and I was here in Puerto Rico.
Yeah. But you got your degree and you'd gone to Cornell and...
Yeah, well, one of the funny things with mycology and with the studies that we do, sometimes our parents don't understand what we're doing. And that was always the issue with with the family, that, oh, you study fungi? Why would you do that? Why would you do that? That's so odd.
When I interviewed at LSU for my job there, I looked at the back row of the seminar room, and here's my father sitting there.
Oh.
And I made the comment, my father is here. He wants to see what my friends and I do, and everybody just killed themselves laughing.
Well, that's kind of the way it was. My father never quite got it. I think he got it a little bit better than my mother did, but she never quite understood how I could get paid to do what I did.
My mother thought I'd be able to go home when my daughter got out of grade school every day.
Yeah.
So, anyway, she really didn't understand. But, well, tell me about, you did go to Miami University, the Cradle of Coaches.
Cradle of Coaches, also a fantastic botany department.
Which still exists?
No.
No,
it's a biology department now. But that was almost by accident. I applied and got into Miami University. I wasn't even sure what I was going to do, but partly because my sister had been a biologist already, my older sister, I thought, this is a good idea. I think I'll do biology. And then I realized there, in those days, I had to make a choice because they had botany, zoology, and microbiology were the concentrations. And so I thought, I'll take botany. I don't like blood, I'll take botany. And so that's how I got started there. And you know about the place a little bit, but they were always known for having master teachers. And they had wonderful people teaching. And I was kind of taken by William E. Wilson, who was plant pathologist, mycologist, is what he taught. And
is that the one they called Doc?
Doc Wilson, yeah. Prof Wilson.
Oh, it was Prof, that's
Prof
right,
Wilson.
You're right.
So, Prof Wilson became my advisor. I took all his courses, and thought, gee, this fungus stuff is pretty neat. And so my junior year, I applied for and got an NSF REU, we'd call them REUs now, undergraduate research experience grant, and went to Cornell, and as an undergraduate worked in one of the labs for the summer. And so that kind of convinced me further, and I was in a plant path lab when I did that. And
Do you remember whose lab it was?
yeah, it was Robert Wilkinson.
Okay.
And he worked on onion diseases. They all have their own diseases
Right.
that they work on. And he was working on Sclerotium cepivorum, which was an onion disease, and he was interested in it because it was a big problem in Egypt.
Oh,
And so he had me growing and working on these things, looking to see if I could discover anything that would inhibit the growth of the fungus. And so that's what I did all summer.
so did you find anything?
No, of course not.
You weren't successful.
That would be expecting too much. He knew I wasn't going to find anything, but
You had to have a project.
well, and he, part of what you do, whether the project works or not, is that you're learning techniques, you're learning how to culture fungi, what you might expect when they grow out. So that I learned all that stuff.
So how did you get mixed up with carp?
Well, I applied for graduate school. I applied to Cornell. I got in. I went to he was in the plant pathology department, the plant pathology department. So within the first year or so, we had to make our choice about where we wanted to go or who we wanted to work with. And Hal Birdsall was just finishing up his PhD with Dick. And Hal had gone to Miami University and had been a protege of Prof. Wilson. And so there were a series of us that Prof. Wilson sent to Cornell. And Hal was finishing up, and I thought, well, he was working on some neat projects and so forth, so I decided I kind of liked the fungi better than I liked plant pathology, so I went with Corf. Yeah.
Good. So then, after that, you got a job here in Puerto Rico?
Yeah, I finished my PhD, and I always say the only job that was available that year was at the University of Puerto Rico. And I had been here, Corf had grants to collect in the Caribbean. And so one of the trips was here to Puerto Rico. So I'd been here, I'd collected, I knew the mycologist in Mayagüez. And so I applied and got the job. And ended up, one of the downsides of the job is that you taught a lot. And so I was here 3 years. I think for the first 2 years, I had like 18 hours a week of contact time. So I was teaching general biology mostly, and then finally I got to teach a mycology course.
And then sometime right in there, you got to go to a meeting at Amherst.
Yeah.
It was the Mycological Society meeting, and I met you at the elevator.
Yes, and
You and Kathy.
my wife Kathy was there, and I remember we rode the elevator up.
You were the first mycologist I ever met who wasn't in the lab at Texas.
Yeah, so that was 1972, we figured.
Yeah.
Yeah. Well,
So we go back a ways.
Yeah, and we remember some of those early meetings of MSA much better than you remember the older ones or the more recent ones. And one of the things that I always remember about those meetings was going on the forays and meeting people on the foray. So, for example, Emery Simmons, who really wasn't a foray guy, but he would go on the foray and he'd be there and
talk to the students.
Yeah, you'd always talk to him. You'd always be able to know that you could talk to him. My first MSA meeting was not that one in '72. but it was 1968.
Wow.
And Emory was president of MSA. And I remember his presidential address, which was Alternaria and Stemphillium, and thinking, wow, these are smart people. They're really something. And he was very dapper. It wasn't the days where you dressed down for a meeting. He was very dressed up, nice shoes. Yeah.
I had made the comment that sometime right in there, Mel Fuller told me that I shouldn't wear blue jeans to meetings.
Well, I might tell people these days they shouldn't wear shorts, especially presenting.
Yeah, I remember the first meeting one of my students went to. He shows up in shorts with some kind of huge necklace on. And I thought, what?
Yeah, well,
but it's changed.
Yeah, it has, but my professor, Dick Korf, was always very casual. And many of us remember the turtlenecks and the peace symbol that he wore around his
That's right,
neck.
I've seen photographs of that.
And he was very much involved in the anti-war movement during my time at Cornell. In fact, to the point that I think his students thought that they were being neglected a little bit. But he was out at demonstrations. He did talks to the SDS, which was pretty radical for a Cornell professor in the ag school to be doing.
The ag school, yeah.
But there was a period there where he really wasn't so engaged in science and doing things. And though he did get this travel grant and so forth to work here. But it was an interesting time there.
Who else was in the lab when you were there?
In the lab was Kent.
Kent Dumont.
Kent Dumont was there and left during my time. John Dixon was a graduate student. Bob Erb did a master's with Dick, and Joanne Rogers, who never really came into the field. She was working on nomenclature with Dick.
Okay.
So it was, Paul Powell. I think I'm
So
the only.
Amy was later?
Yeah. Amy came as a postdoc.
That's true, I forgot about that.
She wasn't a student.
Yeah.
She was Bill Dennison's student.
But then you kept really close to the lab and the people who came later.
Yeah, well, we were always kind of generational, but still knew the.
And Teresita Turiaga was much later.
Yeah, I don't remember how much later. Maybe 5-6 years, something like that. And Linda Cohn was a little bit later.
So then you stayed here 3 years in Puerto Rico.
I did.
And what happened?
And
you were
what
looking
happened?
for other jobs.
Well, I had always said to Dick Korf that I was looking and I wanted to find a spot where I felt that I could accomplish more. And the job at Harvard opened up because I.M. Lamb had retired. And, they never do it this way these days. I never did an interview. I came by maybe in that summer of 1972. I went over to the department, I met people, but I never did a formal interview. So probably the last of a generation who didn't do a real postdoc and who didn't do a regular interview.
And somebody liked your CV or?
Yeah, and they were looking for somebody who could work in the collections as well as teach and do research. And also John Raper was in the department as well. It was the biology department at that point. And John was very anxious that we have a mycology course, and we had planned to teach the mycology course together. And it's one of these great ironies. The week that I arrived in Cambridge, he died. And so we never got to follow
Yes, I
through with
remember it
it.
was some sort of flu-like disease.
I don't really remember. It was an infection of some type. And, it was really quite startling. And I felt that he was going to be my primary mentor,
Yeah.
and then I ended up there without him and without the plan that we had put together. But luckily in the herbaria there was Reed Rollins, who had been director of the Gray Herbarium for a long time, was administrative director of the Farlow at that point, and he was very, very supportive.
What's funny, because I, excuse me, I always thought that John Raper looked so much fitter than his brother.
Yeah, then Ken.
And and it was a shock to me that he died, because he had been in Florida not too long before that, and was very, very charming.
Yeah. Well, you know, both he and Ken Raper were William Weston's students, Cap Weston. And
were they there at the same time?
There must have been some gap between the two of them. I don't remember what their age range was. But when I first went to Harvard, Cap Weston was still coming in occasionally. And, you know, he was such a charming person. He couldn't hear. He had this hearing aid that was a little button in his ear and kind of this box that he carried around. It was very old technology. But he would come in, and he'd often come and chat with me. And the herbarium staff, who could see him in the parking lot, would come. He had a cane, and I see him with a cape, kind of walking through the parking lot. And the herbarium staff would come and say, Don, Cap Weston's coming, Cap Weston's coming. It was just a very
fun. Hide, or
No. They just, it was an event for them too, as much as it was for me. And one of the interesting kind of little research projects that I did involved one of the papers that I found within his files. And it was a fungus that he had collected in Panama, and wrote up a beautiful paper on it, and left it in his files. And this was on bat guano, bat droppings. Phymatotrichum was the genus. And he had written this up. And in the paper, he said that he had collected this, worked on it, illustrated it, took it to Roland Thaxter. And Thaxter said, "Oh, imagine that, you found that thing down there," and pulled out the slides that he had from Connecticut, I think, of this, maybe it's not Phymatotrichum, of this fungus. So we put this together as a paper.
So you have a publication?
Yeah, it was published in Mycotaxon. And so, you know, it was kind of this connection to the past with Cap Weston.
Well, you were in a place where there's a lot of past.
Yeah.
You had lots of ghosts in that herbarium, I guess.
Yeah, well, you know, one of these projects that I'd like to do is to do something, a little book or something that's, my working title is Ghosts, Living with Ghosts, Curator's Life. Because you're working through these specimens, and what you're doing is really kind of recapturing part of these people's activities and lives, so you can tell where they were and what they were doing, and to some extent how good they were with their identifications and so forth. And you know, I always look at these and I think, you know, this is a specimen from Tasmania from 1840. How did they go there, and why were they there? And you know, to think that you're collecting, spending months and months and years traveling around the world.
Well, you know about Farlow, how Farlow did it. Because of the diary. Traveled far away.
No, Thaxter.
I'm sorry, Thaxter. I know better. So talk about Thaxter's diary.
Yes, well, Thaxter did this long trip to South America. He was on a sabbatical in 1905, 1906. And so he traveled to South America ultimately to Buenos Aires, and he kept a diary. The diary begins with his crossing to Liverpool, which is the way you got there. You went from the east coast of the U.S. to Liverpool.
It's like my trip here.
Yeah, and Liverpool across the Atlantic. They stopped in Puerto and various Madeira and various other places, but ended up in Buenos Aires. And part of the deal was that he was going to meet with Carlos Spagazzini. So he wrote about that. Spagazzini had worked on the Label Beniales as Pfister had.
Well, let me interrupt right there, because I just loved reading it. You let me read it the first time I ever visited you. But one of the things I really thought was amazing about Baxter was that he was walking along the street with someone and he saw Spagazzini across the street.
Well, yeah, he was, let me think about that. He was, how was it? He was on the street with one of his contacts. I think it was Davis, who was at the meteorology office. And Davis said, well, there goes Spagazzini now.
Let's go catch him.
And they went over, and Pfister had a reaction to him, piercing blue eyes, walking with a sword cane and an oversized overcoat and a very dirty tie. But he went out in the field with Spagazzini, and they seemed to get along. He went to Spagazzini's house, which is now the Istituto Spagazzini, the collection there.
Well, I just always thought it was amazing that he didn't want to pounce on him and talk to him immediately.
Yeah.
Because that's probably the only man in the world he could have talked to about fungi.
Well, he was a funny guy, I guess. But anyway, on the South American trip, he went around the Horn. He went up to Santiago in Chile. Was very ill for a good part of it, feeling really bad. Then ended up 3 months in Punta Arenas, and the diary was really the inspiration for me to think about in later years of going back to Chile, going back to some of those same locations in Punta Arenas that he had collected, and finding the same fungi that we had in the herbarium from 1906. We could go back 100 years later. I think it was it was about 100 years later. We went back. We found the same fungi that he had collected in Punta Arenas. Yeah, and all of these. Part of it's driven by my interest in cup fungi and ascomycetes, and Pfister had collected many of those interesting fungi there.
I remember the book In Patagonia.
Yeah,
by
Bruce
Chatwin.
Chatwin. Yeah.
Yeah, you told me about that, and it was Chatwin's uncle.
Yeah, his uncle Charlie Millward.
Okay.
And Bruce Chatwin grew up in England, and his uncle, his mother's brother, had been a seafarer of some type, and there was a piece of what was supposed to be a myodon skin in the cupboard in the dining room, and he was always fascinated by that, and that kind of drove him to go to Patagonia and search out the myodon cave and so forth, which you can still go to. But Pfister writes about Charlie Millward in the diary. So Pfister was there when Chatwin's uncle was there, which is such a funny connection.
Yeah.
I don't think anybody else knows that or had made the connection. But it was it's just an interesting piece. I reread that book recently and find it very, very interesting to read it after having been there a number of times. But the fungi are so interesting. We've seen today at a symposium they were doing the Gondwana Reunited symposium, and they're talking about the distribution of these fungi in Australia, New Zealand, and South America. And there were some classic examples like Soteria that my student Chris Peterson worked on. But we have many, many more examples that are involved in that
That's
spot.
funny. I was asking John Pitt about that this morning. You know, the Australian
Yeah.
aspergilli, and did they occur or were they endemic? And he thinks that probably some of those have gone in distribution.
Yeah, I'm pretty sure that they probably do, because that pattern has become, it's just very well established of how that works. So one of the things that Thaxter collected was what we now call Roulandiella, and he called it the little livid pearly white. And that was one of the goals when we went collecting there, along with the fungus that he called geodon, earth tooth. And we collected both of those. Now, they'd both been named, or the underwoody, the earth tongue, the geodon had been named. It's Underwoodia singeri, one of them. And the little pearly white had never been named, but we're now working on this now.
And you have the old specimens.
We have the old specimens, and we have recent collections, and we have DNA, and it's all been kind of put together now as a package. But the only other place those were known to be native is in Australia, where there are a couple species that were described. And I described one from Italy, which I called Roulandiella peregrina for its wandering, because it came with the plants, came with eucalyptus or melaleuca, and that were growing in the vicinity of that spot in Italy. But it's known from Italy, but it's probably Gondwana. It's probably Australia. Yeah.
Interesting.
Yeah.
So are there things that they've left behind? Aren't there some drawings on the wall or stuff from mycologists past? Maybe it's Sparrow. Did Sparrow do drawings? Drawings?
Yeah, Sparrow was a student of Thaxter's, and we do have one of his drawings, which is Christmas in, what is it? Christmas in Fungusville, maybe.
Something like that.
Yeah, and it's all sorts of fungal structures decorated for Christmas. Sparrow was there. Sparrow had this very funny thing about Thaxter. Sparrow was an undergraduate at Dartmouth College, and he came down to see Thaxter to try to get in graduate school. And so Sparrow shows up at Thaxter's office, which was in the Farlow above my office. And he comes into the room, and Sparrow had ridden the train down in the smoking car, and he comes into Thaxter's office, and Thaxter says, the nicotine.
Did he smoke?
No, but he was in the smoking car.
No,
Thaxter, he was not one with vices.
Yeah,
he didn't like it. But Sparrow was good. He told stories. One of them was his thesis was on monoblepharidales, or monoblephars, and Thaxter had collected and described some of these things. And Sparrow began his PhD work and couldn't find it. Couldn't find monoblepharus. That was what he was supposed to do, but he couldn't find it. So he finally goes to Thaxter and says, "I just can't find this." And Thaxter said, "Well, you take the trolley out to Arlington, to the reservoir, walk along the kind of south side of that, and there's some sticks. And if you pick up those sticks, you'll find it." So he hadn't been there in 40 years probably. But Sparrow went out, he went, looked at the sticks, there it was. So, it's amazing. Yeah, very funny.
So you continue on to the future, but you seem to have lots of international students that come in and gives them a
these
really
days,
nice experience.
we've had In the last couple of years, we've had a number of students from Pakistan, and mostly women, and I've been very happy to have them in the lab because I think they are improving their skills and training by being here. And none of them, one of them perhaps had traveled, but none of them had been outside of Pakistan before they showed up in the lab. And they've been doing sequencing and phylogenetic work, mostly with agarics, but they've been very productive in their time there. We've had, with the lab rules, we've had a number of visitors who have come through from Poland and Hungary to work on lab rules recently.
Italy, Rossi.
Italy, yeah. Walter Rossi has come and visited, and he was very interested in some of Thaxter's line drawings of unillustrated lab rules. But so the future. Where's the future? I've spent my life in the herbarium and working with specimens. We're now seeing a kind of resurgence of interests in herbaria because of the information that they hold, with label information and distributional information. We've been working on a project with digitizing rust specimens and collecting data on our rust collection, which is quite big, quite large. So I think that there is that aspect, but there's also the aspect of how these collections are documenting the world. And we know now, at this conference, somebody was just telling me, I'm trying to collect cordyceps, and I go to the locations, and there's now a city. There are things built over it. Yeah. So the future for collections is that they hold this vast diversity that is going to be tapped in one way or another, whether it's collection data, sequence data. That's going to really be an important thing. We just did a little manuscript on techniques to look for endophytes using high throughput sequencing in herbarium specimens. So taking these old specimens and being able to pull out the fungi that are in the specimens, and then you can kind of open this up to say, well, you know, if you're in the same location with the same plant over a period of 100 years, has that endophyte community changed, or has it shifted? And so, you know, without the herbarium collections and without these materials, you don't have a chance to do that. You know, it would be incidental to find old material otherwise. Pillow stuffings or something. You know, you'd have to be really inventive to come up with some of this. So I see the future as being continuing to document. You know, thinking about this Patagonian work, you know, now we have much better collections than ever existed for Patagonia because we've been there and been working on it. Do we know what Patagonia is going to be like in 15, 20, 50 years? No, it's going to be different. But we have the material now that shows us what it was like. Not very happy.
Sad.
Little pessimistic there. Sorry. Don't you have anything else you want to be upbeat on?
Upbeat. Okay. So let's see. So what are your plans? Are you planning another trip somewhere?
Sure. I'm going to do a trip to Panama and Costa Rica in February. Not collecting so much, but with a Harvard group, and we're talking about trying to develop a project to do a little bit more in Chile.
Oh, good.
And what the nature of that is, we don't know. You know, we've collected, and mostly, you know, Matt Smith and his group have led that, but it's been collected very intensely now, at least for certain seasons. And what we would hope to do is to be able to figure out now, you know, can we do some gradient studies? Can we do some seasonal studies and collect? We've missed a lot of early spring, I think, with our collecting pattern. We've got a little project on the morcellas in Chile that we're thinking of working with, because those are now economically quite important for field harvests and so forth, wild collected morcellas, big export for Chile. But some of them that are stimulated to fruit by fire have encouraged people to burn more than they should burn.
Yeah.
And so we're trying to think about what we can do if we can understand the biology of these a little bit better. Can we do anything about this? Alerting people to the idea that they can collect these without burning. So anyway, there are a number of those projects.
That is a much more optimistic.
Yeah, thinking of ways in which we can use our knowledge to preserve and conserve is part of what we should be doing.
Yeah.
Yeah. Yeah.
Thank you.
Okay, thank you. Pleasure.